Our laboratory at the University of Dundee, based at the James Hutton Institute, studies barley, Scotland's main cereal crop. Barley is used for animal feed as well as in the brewing and malting industries, making it a vital part of our economy. We investigate how barley grows and how its grain develops. We do this by studying its genes, the genetic instruction manual that makes barley, barley. To better understand grain development, we want to examine the genetic profiles of individual cells within a grain. To separate out these cells, we use a technique called protoplasting. In our glasshouses, we grow the plants we need for our experiments. When the plants are old enough, we harvest the tissue we need and take it back to the lab. Once there, we dissect the grains and put the tissue in a dish containing a washing solution. We then dice it up into very small pieces. When we have all our samples prepared, we remove the washing solution and add an enzyme solution. The enzyme solution breaks down the cell walls that surround plant cells, so that individual cells can float freely in the solution. To let the enzymes do their work, we put the samples on a shaker and leave them for several hours. Sometimes the enzymes need help to break down the cell walls, so we gently pipette the samples up and down a few times to help release the cells. We then place the samples back on the shaker. We repeat the step as needed to make sure the cell walls have broken down. To remove any remaining debris or big chunks of tissue, we filter the samples and then pour them into tubes. From here on, to keep ourselves alive during this process, we take care to keep them on ice as much as possible. To get rid of the enzymes in the solution that we don't need anymore, we spin the samples in a centrifuge. Here the cells will fall to the bottom of the tube and form a pellet. We remove the liquid above the pellet, called the supernatant, and add clean liquid to the sample. We want to make sure the samples are as clean as possible, so we break up the pellet, allowing the cells to freely float in the solution again. We filter the samples to remove any remaining debris and spin the samples in the centrifuge once more, slower this time. Again, we remove the liquid in the tube, leaving just a little bit for our cells to be mixed into. We filter the sample one more time and put it back on ice. To check if the protoplasting procedure has worked and find out how many cells we have in our sample, we look at them under a microscope using a special microscope slide called a hemocytometer. We add a dye to the sample so that cells that are alive stain green and dead cells stain red. We can then count how many of each are in the sample and calculate the concentration of cells in the sample and what percentage of them are alive. Finally, we examine the live cells to discover which genes control grain development. This knowledge may help make better barley that produce more grain.